All right, so I'm going to expand on chapter 17, which is all about gene regulation. And we're going to focus in on transcriptional control of gene regulation. And so, as we talked about before, you can have transcriptional regulation, post-transcriptional regulation, translational regulation, post-translational regulation. You can have regulation at lots of different levels. But we can get into the details of transcriptional regulation, and this is a few slides I've put together from some different things. A few, as in 63. So this is going to be a longer video, and it's also I'll probably break it into two. We'll see how it goes. All right, so I like this next figure here. This really summarizes what's going on with gene regulation at the transcriptional level. You start with chromatin, which is condensed, so condensed chromatin. And there are ways to open and close the chromatin. There are protein activators that can actually bind onto chromatin and cause it to open up. So there are activators that cause it to open up. There are repressors that cause it to tighten up and turn the gene off. There are also things to open chromatin up are things like histone acetylation will open up the chromatin, and histone methylation will tighten it. So this is stuff on this side is going this way and condensing the chromatin, and stuff on the other side is going this way and loosening up the chromatin. So there are other things that can do this, but we're going to focus just on these. We might as well throw in DNA methylation here as well. That can be a point of confusion. You just have to make sure when you're talking about methylation, are you talking about the actual DNA that gets methylated, or are you talking about the histones that get methylated? Either one of those can lead to histones going in this direction, or the chromatin going in that direction. Once the chromatin is open, then there are certain cis-acting sequences, and you can't really see them here, but they would be underneath these proteins here. So there'd be little boxes like enhancers that are being bound by these various proteins. And so these would be activator proteins. And you see that there's part of the activator protein that binds the DNA and part of the protein that interacts with the mediator. This mediator is the thing I like to call the mega blob because there's just tons of proteins in there. These proteins are called co-activators. Together they interact with the general transcriptional machinery. So all these little proteins here are things like TF2A, 2B, 2D, etc. Probably this one, though they don't have it labeled, but that's probably part of TF2D. And remember, part of that is the TATA binding protein. So there are a lot of abbreviations in this field of molecular biology. So you just kind of have to get used to that. And so that's kind of how things go. And so what we want to talk about in this chapter is some experiments that led to this model. So this is a model. So a model is sort of like a complicated hypothesis. So this is sort of a framework for how we think that genes are regulated. And all of this is supported by various kinds of experiments. So we want to talk about some of the experiments, the types of experiments that you would do to test this model. All right, so just to summarize, we got loose chromatin. Well, we start out with tight chromatin. And then we got stuff, well, we'll do it this way. We got stuff that loosens it up. And there's a transcription start site there. There are some sequences close to the transcription start site. There are some sequences far away. This might be an enhancer. Now there are other things that there are lots of different sequences, and we can expand exactly what's in these sequences. but this just gives us the general idea. Then we have activator proteins. Those activator proteins cause the DNA to bend. So now we got this activator protein. It interacts with the mediator, which interacts with the general transcription apparatus, general or basal transcription apparatus. And then that'll act with, what's good color for RNA polymerases? Go here and get some purple. That'll interact with RNA polymerase. And mainly we talk about RNA polymerase 2. And in here somewhere there's a transcription start site, and RNA polymerase will start to transcribe the gene. So how do we know about these things? That's what we're talking about. All right, so these are the things we have to talk about. And even though sort of in the order that they work in the cell, when a gene goes to be turned on, well, it's not exactly clear exactly what order they work in. But this chapter is organized sort of historically in terms of what kinds of things were discovered first. And so we started by discovering cis-acting sequences, then looking for transacting proteins that bind those sequences, and then later, even though this may happen first in some cases, what kinds of changes in chromatin structure occur that cause genes to be expressed. And a couple things we'll talk about are histone modification and chromatin condensation, decondensation. Something else that should be in here that is very much related is DNA methylation also causes condensed chromatin. But again, that's DNA methylation, not histone methylation. All right, so let's get going and talk about some experiments. So when we're talking about gene expression, we're mainly, especially when we're talking about transcriptional gene expression, we want to be able to demonstrate that some genes are expressed some of the time. So here's a gene, and let's say here's another gene over here, and both these genes are in the same cell. And let's say this is a liver cell. And let's say that this gene is expressed in the liver. So this is RNA, and this gene is not. So that gene's expressed somewhere else. So that we call that differential gene expression. Just simply gene that is expressed differently or differentially in different cell types. And there's lots of different situations that might make one gene be expressed and another one not. This simply, we've got a liver cell, and this gene, whatever it is, makes a protein, and that protein does some kind of liver-specific function, whatever livers do. So livers have a lot of enzymes, metabolic enzymes, whatever. You need that protein in the liver. So you have to turn that gene on in the liver. So the way that works, generally speaking, is that there's some DNA sequence that's present in front of that gene, but not in front of this other gene on the left. There's a liver-specific protein, transcription factor, that binds that DNA sequence and turns the gene on in the liver. And then when it does that, all that other stuff happens too. All the basal transcription apparatus and all that kind of stuff also happens. So if we want to demonstrate that indeed some genes are expressed in some tissues and not others, we have to do an experiment. And here's an example of this type of an experiment. So let me go back for a second. So if we want to detect this, we can detect different DNA sequences by making what's called a probe. So if we make some kind of a probe, this will make it green. So we're going to take a piece of this DNA, and we're going to cut it out using restriction enzymes, or maybe do PCR and amplify up that region of DNA. We're going to radioactively label it and then make it single-stranded. Then we can hybridize it. So we're doing all this like in a test tube or something, or on a gel. We can hybridize that DNA to the RNA. So this is called hybridization. And because this is radioactively labeled, wherever that probe, and probe's an important word, that probe will stick to that RNA, it'll show up. That's radioactive, and we can expose film and get a dark spot whenever that probe sticks. So this is a different type of experiment, but it's still a hybridization experiment. The importance here is that you can always use hybridization to detect RNA. And when we're looking at gene expression, we want to detect RNA or protein, right? Because those are the products of gene expression. So that means gene expression when we go from DNA to protein. And we can measure the RNA, or we can measure the protein. It's somewhat easier to measure the RNA because it's easier to get a probe for the RNA than it is to get a probe for the protein. So here they've done this in a slightly different way. Trying to figure out how to make these. So if we look at these, on each of these pages, there's notes. And so you could read this, you want to go over this PowerPoint, listen to what I say, but you can also read these notes. I'm going to talk about the different kinds of blots here in a minute. Okay, so we're going to go, we'll go back to our normal view. And in this case, what we've done is we've put DNA, so cDNA. Let me go back a step. So this RNA, if we want to study this RNA, one thing we can do is we can take that RNA and we can use what's called a primer, just like in DNA replication. We can use reverse transcriptase and convert that RNA into DNA. So that's reverse transcription. This reverse transcribed DNA is called cDNA. It's a copy of the RNA, so it's called cDNA, C for copy. So we can take those cDNAs that correspond to specific genes and put them on a piece of filter paper. So that's what that is. Then we radioactive, so on that paper, you've got DNA sticking up, and each DNA is from a different gene. And then now we're going to radioactively label the RNA, so it's a little bit backwards from some techniques. And if that radioactive RNA sticks to that spot, that means that RNA is present in the sample. So we isolate RNA from these different tissues, and then we hybridize them to these different genes. And when we see a dark spot, that means it's highly expressed. It just means there was a lot of RNA in that sample. And if you see something like that, I don't think there's anything there, but something like this, that's no expression or very low expression. Now these controls, these are genes that are expressed everywhere. And so you always want to have some kind of control like that so you can see what's going on. So what do we conclude from this? We've got 12 genes that are spotted on this piece of paper, and then we hybridize radioactively labeled RNA to it, and wherever we get a dark spot, that means the gene's expressed. So all of these genes are expressed at some degree, or to some degree, in liver cells, but not in kidney cells. You get a little expression here and here. You get very, very little expression in the brain. These are some control genes that should be expressed at least a little bit everywhere. And so what we see is that all 12 of those genes are expressed in the liver at higher rates than they're expressed in kidney or brain. So that's what we mean by differential gene expression. The same gene. Each of these things has the exact same gene. So that gene is here, that gene is here, that gene is here. But it's only expressed here. So if we looked at one particular gene. Gene is present, but only expressed in certain tissues. All right, so why is that? So it's sort of like when we looked at the lac operon. We had the lac operon that was only expressed under certain conditions. No lactose, the gene was not expressed. Plus lactose, the gene was expressed. When we studied that, we realized that the reason for that is that there are cis-acting sequences that control expression of the lac operon, namely the operator. And then there's a transacting factor, namely the repressor, that binds the operator. And those two things control whether or not the gene is expressed. So when people started studying eukaryotic genes, they started saying, well, maybe there are cis-acting sequences that control eukaryotic gene expression. So they looked to study this. They did a couple things. One, they started looking at genes. They said, okay, we got a gene. If there are cis-acting sequences, they're probably up here somewhere. So this is called the upstream region. And so we can go in there and we can look and say, are there any DNA sequences up there that control expression? One way to do that is to mutate those sequences and see if expression goes down. So if there's some sequence here that's necessary for expression, then when it's present, you'll get expression. But when you mutate it, you'll get no expression. So that's one type of experiment. Then another type of experiment is say, well, let's look at a bunch of genes. So if there might be some sequences in common to all genes or lots of genes that control expression. So let's compare these sequences and just see if there's any common sequences that are found everywhere. And just to give you, sort of a prelude, guess what they found? They often found in front of lots of genes this TATA sequence. So that's one way to find it, is to compare a bunch of sequences and identify what's called a consensus sequence and say, okay, well, that consensus sequence, because it's conserved, if it's in lots of different genes, that means it's conserved over evolution, and that means it must be important, or it leads to the hypothesis that it's important, and then that leads to say, okay, well, so that gives us a target. Let's mutate that and see if anything happens. All right, so let's look at how these experiments work. Here's an example of some mutants. Don't worry about how they made them. They use a technique called linker scanning. But all that means is that they inserted sequences in different places. So they're just inserting a mutation in different places. And then they measure gene expression. And they're just kind of being not really quantitative here. They're just saying we get a lot of expression, or kind of a little bit of expression, or we get no expression. And so if we look at, we can mutate different sequences. So we're going to mutate different sequences up here. And then we're going to measure gene expression. And if gene expression goes down, that means we've mutated something that's important for gene expression. All right, let's say you've got a bunch of light switches on the wall, and you want to know if any of those light switches are responsible for specific lights in the room. The way you would test that is you would start to mutate those light switches or turn them off and see which lights go off. Now here, there's lots of different ways to measure expression. And so here they're going to use a reporter gene, and I'll talk about that in a minute. But there's lots of different ways to measure gene expression. So however you measure it, you come up with some relative level of gene expression. All right. So if you don't mutate anything, you get, this is your maximum amount of gene expression. Here we've mutated something down here, but we haven't affected expression. So that region is not important for gene expression. Here we've mutated something and we've decreased expression. So there's something there that's important for maximum expression. We mutate something here and we also decrease expression. So there's something there that's important. We mutate something down here and we completely abolish expression. And so each time we hit something that is important, we say that is an element or a control element or a cis-acting element that's important for regulating gene expression. All right, so in this case, they've identified 3 elements and they don't really have them lined up, right? So they're kind of expanding this up. And they've got the top box, which is an important element. And if you don't have that, you get 0 expression, so that you need that for minimal expression. And then these are elements that are in the promoter, and their PE stands for proximal. So that just means close to the promoter. So proximal element 1 and proximal element 2. So that's just how you identify cis-acting sequences. I want to take a little bit of a side tangent here to talk about how you measure gene expression. So we just talked about reporter genes, but let me first, and we talked a little bit about dot blots, but let me just first talk about blots. So a blot is you isolate something, you run a gel, and I'll talk about that in a minute, you transfer or blot that something to a filter paper, then you hybridize to a probe. We will do one of these types of blots, maybe using the CASEIT software. We'll see if we have time to sort of illustrate it. All right, so now if the thing that you isolate is RNA, so if you isolate, and so always wait, so before we do that, so then what you'll see is if you run a gel, you'll have all the different molecules present. Transfer to a filter, and you can't see anything yet, and then hybridize to a probe. Once you hybridize to a probe, only the thing that you're looking for shows up. So here, this is all, let's say we're doing RNA. We isolate RNA, it's a northern blot. And so all the RNA shows up, but the RNA that we're interested in is only the RNA from a particular gene. So we're only interested in that RNA. So we make a probe, which is a piece of DNA, or it could be RNA, but a piece of DNA that sticks to that RNA. We radioactively label it, and when that probe sticks to the RNA, it shows up on this blot. So this is a northern blot. So that's one way to measure gene expression. Now you can do other kinds of blots. But you have to be careful because only some of these are for gene expression. So we're going to look at a few types of blots here. So we're going to run something on a gel, and we can run DNA, RNA, or protein. If we run DNA, it's called a southern blot because it's named after Ed Southern, the guy who discovered it. If we do RNA, it's called a northern blot. It doesn't get capitalized because it's not named after somebody. If we look at protein, we do it's called a western blot. And then we probe with, you can probe with a DNA probe or an RNA probe, but DNA will stick. But when we do protein protein is a three-dimensional thing of amino acids. Up here, the probe sticks to the DNA by hydrogen bonding in between, like, so if we have a particular sequence, the probe would match up to that sequence. So that would be our probe. But a protein has like methionine, arginine, serine, threonine. Those are not bases. You can't base pair to those amino acids. So we need to do something else. So we make what's called an antibody. And we can radioactively label the antibody or fluorescently label it. And so the probe when you're doing a Western blot is an antibody. These can be used to look at gene expression because we're looking at differences in whether or not the gene is expressed. So do we get more RNA or more protein? The DNA is always there. So if we look at DNA, we're not looking at gene expression because the molecule DNA is not what is expressed. What gets expressed is RNA and protein. So it's very, very common when I say, tell me an experiment to measure gene expression, a lot of students will say a Southern blot. That's not a good answer because Southern blots look at the DNA itself. You can look at changes in DNA, but you can't look at changes in gene expression. Okay, so some other types, so that's a Northern blot. A dot blot is the same kind of thing, but you just don't run a gel. You hybridize RNA and DNA on a filter without running a gel. A Western blot is for proteins. Microarray we're going to come back to later. A microarray is a lot like a dot blot. It's just a slightly different technology, and you have a lot of pieces of DNA. So you just have this little chip with lots and lots and lots of spots. So it's just like a dot plot, but it's like on a glass chip instead of on a piece of paper. And each of these has a little piece of DNA in it. And then you can radioactively label your RNA, and wherever the RNA sticks, then that will light up, and that will show that that gene is expressed. We're going to talk more about that with the KSIT experiment, so we won't, and there's videos on microarrays. Now so the difference is, so these guys only are looking at one or a few genes at a time. And this is looking at thousands. Okay, so we'll come back to that. But let's talk about reporter genes real quick. So now let's say we have some suspected, we have some sequence up here. We want to know if this sequence contains any sequences that will drive gene expression. And this is just whatever, gene X. This is the actual gene that we're interested in. But rather than measuring the RNA that gets expressed from that gene, you can take these control sequences and connect them to some other gene. And this gene is called a reporter gene. And that just means that's just some other gene that is easy to measure the outcome of. And there's several different examples. For example, beta-galactosidase. You can use that as a reporter gene, and that takes X-gal and cuts it into X plus gal, and the gal part, or actually the X part, is blue. So you can measure. Excuse me. You can measure how much blue stuff is made, and that tells you how much the gene is expressed. There's another one called luciferase, which is kind of cool. and that measures light. And so luciferase comes from firefly butts. So when fireflies light up, they light up because they're expressing luciferase. And so you can measure whenever that gene is expressed, you can measure how much light is emitted, and you can use a little light detector to measure that. And that's a really good way to measure gene expression. There are others, but those are the examples. So reporter gene is just something that's easy to measure. It's unrelated to the actual gene, and so then you can take your different sequences and mutate these different sequences, and then measure how much reporter gene product is made, and go from there. So let's take a little sidetrack and look at Google up reporter gene. Let's look at luciferase. And let's look at some images. All right, so this is showing you how luciferase works and how you would, so here's a good picture down here. You've got a promoter connected up to a reporter gene. It gets transcribed into messenger RNA, and then it makes some kind of a signal. I just wanted to see if we have a good picture of something that's actually lighting up. So here's some light coming from some organism that, oh no, those are on a plate. Those are colonies on a plate that are expressing luciferase. Here's a plant that it's expressing luciferase. Etc., etc. There's a northern blot. So anyway, it's probably not a bad idea to look at some of these things. Another important reporter gene, I almost forgot about, is green fluorescent protein. But we don't need to have luciferase at the same time. Green fluorescent protein as a marker for gene expression. That's exactly what we're looking for. Let's see some pictures. Ah, look at that. That's beautiful. So here's some mice that are expressing green fluorescent protein. Here's some bacteria that are expressing green fluorescent protein and blue fluorescent protein. Here's some people that made a nice little diagram with bacteria that are expressing different colors of fluorescent proteins. So that's what we mean by reporter gene. Here's probably blue fluorescent protein. There's lots of different ones. Here's jellyfish. This green fluorescent protein originally came from jellyfish. Here's a mouse that's expressing a gene only in particular places, in the ears, in the nose. This might be something that's controlled by temperature. Sometimes genes in the periphery get turned on because the temperature is lower. Here's a cool picture on the cover of Science. This is Caenorhabditis, which is a worm, showing expression of the gene here. Here's an actual picture of the green fluorescent protein itself. So green fluorescent protein is a very useful reporter gene. All right, so we got that reporter gene. So we've got luciferase beta-gal GFP, which is green fluorescent protein. So somehow easy to measure. You can just measure the intensity of expression. All right, so we talked about reporter genes. All right, so another way is to look for consensus sequences. And so we can, I'm going to back up one minute because there are a couple of other techniques I want to talk about. And we already talked about this. But there's a couple of other ways to measure gene expression. So, and these are more common these days. So here's some RNA. You can make a primer and use reverse transcriptase to make a copy of the DNA, and then do PCR and amplify up that DNA a whole bunch of times, and then measure it. So this is called this part is reverse transcriptase. And so this is sometimes called reverse transcriptase PCR, RT-PCR. And if you do it in a quantitative way, it's called quantitative PCR or qPCR. And now one more way to measure things is by a technique called RNA-seq. Now this is more of a genomic technology, so we'll come back to it in the chapter that's on genomics and proteomics. But the idea is here we have a bunch of RNA, and we convert it to DNA by reverse transcriptase, and then if we did PCR, it would be called RT-PCR, but if we just sequenced it, it's called RNA-seq. And we don't have to worry about exactly how you do that, but there's new technologies that allows you to sequence RNA at a very high level. So we will come back and look at RNA-seq, but I just wanted to point that out. So the old school way is a northern blot. You can do western blots as well, but we're just talking about RNA now. You can do dot blots. These are kind of super old-fashioned. Microarrays, if you want to look at thousands of things, that's a little bit old-fashioned. But now what we would do is look at qPCR or RNA-seq. These are the new ways. And all these things is measuring gene expression. What isn't it? It isn't ever measuring DNA itself directly, because all this stuff comes from a gene. So this is DNA. The DNA stays the same, but the RNA gets expressed. So one way that cis-acting sequences upstream of genes have been found is by what we just said. Take a cis-acting sequence, connect it to a reporter gene, measure expression. Another way is to compare a bunch of different genes and look for a consensus sequence. So this is where I said, well, let's take this gene, and this gene, and this gene, and compare the sequences upstream of their transcription start site and see if we find anything in common. When you do that with a lot of eukaryotic promoters, what you find is a consensus sequence called the Tata box. So in 91% of the genes, there's an A in this position. In 97% genes, there's an A in that position. 95% there's a A here. 100% there's a T there. 83% there's a T here. So this is a consensus saying here, it's either an A or a T. Here it's either an A or a T. But not always. These are just sort of give you a general pattern to look for when you're looking upstream of genes. Say, does it have a Tata box? Okay, so that's identifying cis-acting sequences. Here's a picture of how another example of mutating the upstream sequence and then looking for the cis-acting sequences. So here's the upstream region. Now we're going to make mutations rather than all those insertions. We're just going to cut off a little bit in each case. Cut off a little there, cut off a little more, cut off a little more, cut off a little more. Then we put these things into a plasmid so that they're upstream of a reporter gene. Put those into cells, and then if the gene gets transcribed and translated, then we can measure this reporter gene, the product of the reporter gene, and then we can get a readout of how much gene expression there is in each case. So this is just showing making the deletions. So we're deleting a bunch of we're deleting this upstream region. Whoops. We're deleting this upstream region, and then we're just going to see if there's an effect. If transcription goes down, that means there was something there that was important. Put it into a vector, so you have to cut and ligate things into plasmid vectors. I don't think we're going to have time to go over that in a whole lot of detail, but you certainly did that in previous classes, and so you should, and you're going to read the chapter, so you should have a general idea of how you cut DNA and put it into a vector. You put that vector into a cell. We can call that transfection. And then you measure the reporter enzyme. Maybe that's light. Maybe that's GFP. Maybe that's beta-gal turning X-gal blue. Right, so those are 3 different types of reporter genes. So in this case, plasmid 1 and 2 had no effect. Plasmid 3 and 4 decreased it, and then plasmid 5 eliminated expression altogether. We put that together, plasmid 1 and 2, no problem. Well, so this one, when we have the full sequence, we get full expression. If we remove a little bit here, we still get full expression. So there's nothing all that important here. There's nothing that's important for gene expression there. But if we delete this part, we decrease expression, which tells us that there's something there that's important for expression. We don't decrease it any further if we delete this part, so there's nothing important there. Here, we wipe it out. So by doing that, we've identified 2 different elements that are upstream of this gene that are necessary for transcription. When you do this and you look at a lot of genes, you start to find that these things are pretty complicated. And they're more complicated in mammalian genes than in yeast genes. So yeast is a simple microorganism, but it's eukaryotic. And it's just simpler. So this is just kind of giving you an idea of what a typical gene looks like. So in mammals, you have transcription start site. You have a ta-ta box. You've got some proximal elements, and you can have a bunch of those. So that's what these guys are. You've got some sequences that are far away. Those are enhancers. In yeast, they call them a UAS, or upstream activating sequence. And the thing about enhancers is enhancers can be found anywhere. You can actually find an enhancer in an intron. You can find enhancers downstream of genes. So you can find, that's kind of a definition of an enhancer, is that it's a sequence that can affect transcription, and it can do so whether you put it upstream or downstream of a gene. They can be in introns. So they can actually be kind of hard to find. I mean, if you're looking for a promoter element, you know that's going to be within a couple hundred base pairs of the start site. If you're looking for an enhancer, it could be anywhere. And that makes it really hard to find them. And sometimes you'll get a mutation in an enhancer that causes a disease, and it's really hard to track it down. Okay, so what we've done so far is talked about how you identify cis-acting sequences. Now we want to identify how you find the proteins that bind them. What kinds of transacting factors do we expect to find? Well, RNA polymerase, of course. Basal transcription factors like TF2A, B, D, etc. Regulatory transcription factors like activators, repressors, repressors. Co-activators, etc. And co-activators are part of the mediator, or the megablob. All right, so how do we do that? There's a couple ways to do this, and the first thing is to look for DNA binding proteins. And one method is called DNase footprinting, and one is called a gel shift. Officially, this one's called electrophoretic mobility shift assay. This DNase footprinting is going to be very similar to what we've talked a little bit about in terms of DNase hypersensitivity, but this is a little bit different. Here we're looking for DNA and a protein that binds it, we're not really looking at chromatin. So let's look at some of these experiments. So this is a footprinting or a DNA protection assay. So you have a DNA sequence. You've identified a cis-acting sequence somehow. You have a protein, so you have two test tubes. One test tube has the DNA in it, and one test tube has a protein in it. And we're going to mix them together. And one of two things can happen. Either the protein binds the DNA or it doesn't. If it binds, it looks like this. If it doesn't bind, it just kind of looks like this. And so we can figure this out. So we take this DNA and we treat it with DNase, and DNase will chop up the DNA and it'll cut it in various places. But if there's a protein bound to the sequence in question, then the DNase can't bind here. So DNase is an enzyme and it's trying to bind here and cut, and it can't do it. So whenever it cuts, we've got a radioactive label here. And so this band right here will show, we're going to run a gel, and these bands will show up on a gel. But there won't be any bands that correspond to this region. Over here there will be. Over here we'll have a band here, we'll have a band here, we'll have a band here, band, band, band. We'll have bands everywhere. But here we'll be missing bands that correspond to this size. And that's because this is protecting the DNA from DNase. It's sometimes called a DNase protection assay, but it's usually called footprinting. And the result looks like this. So let's just look at two lanes. So this would be DNA that doesn't have a protein bound, and this would be DNA that does have a protein bound. And the protein is bound right in this region. And the reason is because in that region, the DNase can't get to the DNA to cut it. So you don't get any bands that correspond to that size. And so you get a blank space in the gel. This particular experiment, they're running a bunch of different samples, and each of these samples all has protein that binds that particular DNA, and all the other samples don't. So if you just compare these two, you can see what it looks like, and this protected area is called a footprint. Now another one that I think is even easier than DNA footprinting is we're going to do the same thing. We got DNA in a tube. We've got protein in another tube. We mix them together, and if the protein binds, it looks like that. If the protein doesn't bind, it looks like that. So now we just run a gel. We have to radioactively label the DNA. And so if there's places where the protein doesn't bind, let's say we have we just take this DNA here and we load it into the first lane on the gel. Remember, DNA runs from the minus to the plus lane. Well, this DNA is going to all run down here. So it's a pretty small piece of DNA, so it runs a long way. In the next lane, let's say there's some protein or some DNA, and we add the protein as well. And let's say that protein is there. Now in this case, there's a lot of this DNA, so not all of it is bound to that protein, but some of it is. And when the protein's bound to it, it runs slower. So the reason for that is because the DNA in here is bound to a protein, and it runs slower. And that's it. And so let's say, well, what about this protein? Does this protein bind? Let's say there's another protein. That protein doesn't bind. So you'd say, nope, that protein doesn't bind. So that's a gel shift. It's also called an electrophoretic mobility shift assay, which is a long word. But this type of technique is called electrophoresis. Mobility means movement through the gel. Shift means that we're shifting the mobility, making it run slower. And A means assay, which is another word for like just experiment or test or technique. So this is what they really look like. Here's a whole bunch of samples. And which samples do you think contain DNA bound to a protein? So here's our DNA without a protein. What do you think is going on here? That's DNA with a protein. Gel shift. Shifts where that DNA is running in the gel. Things are running this way. These things are small. These things are bigger. Bigger is slower. Think about trying to run a foot race and then trying to run a foot race with me jumping on your back. You will run slower. All right, so I would be the protein, you'd be the DNA. All right, so that's how transcription factors biochemically are identified. Take a DNA sequence that you think is important for controlling transcription, mix it with a protein, run a gel. If there's a gel shift, then the protein has bound the DNA. So when I ask you to tell me about an experiment that could be used to demonstrate that there's a protein that binds DNA, say gel shift. I'm going to explain how you would do it. And I'm not worried about all the technical details. I'm just worried about sort of a general flow of how you would do it. So now we're talking about experiments that led to the idea that if we take a protein, a transcription factor, and so we've identified a protein. Now we've cloned the gene for that protein. So now this is our transcription factor. And this is our cis-acting sequence. And now we want to ask, if we make this protein, can this protein bind on here and activate transcription? So here we asked, so what are we asking here? Does protein bind DNA? Just because it binds DNA doesn't mean it activates transcription. It just means it binds DNA. In science, you have to ask each question specifically. You can't say, well, we know that protein binds DNA, therefore it must activate transcription. No, because it might turn off transcription. Who knows? It might have nothing to do with transcription. And so this is saying, does protein activate transcription? Now we would expect that the protein has to both bind DNA and activate transcription, and that's usually the case. Not always. All right, so here what we're going to do is we're going to express this protein. We've got our cis-acting sequence connected to a reporter gene. We're going to put both of these plasmids in a cell. And if this protein, so this is making a protein, and then this protein is going to bind to this DNA, and if it activates transcription, then we'll be able to measure the outcome by measuring the expression of the reporter gene. So now we're going to look at specifically the protein that binds. So we're doing an experiment where we're going to express a protein. In this case, there's a protein called GAL4, which binds onto this enhancer called UAS-GAL. It's just a classic example. And we're going to use the LAC-Z gene as our reporter gene. So this has nothing to do with bacteria or anything. It's just happened to be taking the LAC-Z gene from the lac operon and using it as a reporter. When this gene is expressed, it makes beta-galactosidase. Beta-galactosidase normally breaks down lactose, but here we have a specialized substrate called X-GAL. When we break that down, it turns blue. Don't believe me? You probably believe me, but let's look it up. Not Lacey reporter. Boy, that could get, let's see here. LAC-Z reporter images. let's see. Oh, here's a good one. I like these. These are Drosophila embryos with the LAC-Z gene expressed in a particular pattern in early embryos. Here's some more Drosophila embryos showing that the LAC-Z gene is expressed in certain areas. Here are some graphs so you can quantify LAC-Z expression or beta-galactosidase expression, measure it, and put it on a graph. And here's a case where it's expressed a lot, and here's expressed a little. I like these pictures being expressed in Drosophila embryos. In mouse embryo, there's a little LAC-Z reporter gene being expressed in the hindbrain. Here's some in bacteria. I think that's bacteria. It could be something else. Some more mouse expression, more Drosophila expression. Here's a picture of a vector that you could use to express it. So you get the idea, right? So it's blue. When the gene's expressed, it's blue. All right, so that's the reporter gene we're going to use. And now, this looks a lot like that other diagram, but now it's a little bit different. So now we're going to take the protein, this GAL4 protein, that we showed has showed binds DNA. It binds the UAS GAL. Let's say we did a gel shift and we know that it binds that. So these results are from gel shift. So how do we know whether it binds or not? We do a gel shift and say, yep, okay, it either binds or it doesn't bind. And then we can measure beta-galactosidase activity by using xGAL and seeing if it turns blue. So this is this experiment. I'm going the wrong way. We're taking the protein, so GAL4 protein, putting it here, expressing it, and asking if it activates transcription of a reporter. And that's the reporter. And so here's protein. How do we know it's protein? Look at that. They gave us an N-terminus and a C-terminus. Remember that? If we express the entire protein, it can do both things. It can bind DNA and it can activate transcription. If we mutate this end of the protein, it can no longer bind DNA. And if it can't bind DNA, it can't activate transcription. So that makes sense. If we mutate this end, it can still do both. It can still do both. Can still do both, but not quite as good. Can still do both, but not very good at all. Can't activate transcription. It can still bind DNA, but it can't activate transcription. So what these experiments tell us is that down here, there's something that's needed for DNA binding. And down here, there's something needed for activation. So we can separate the functions into different domains of the protein. And so after they do a whole bunch of experiments, they can narrow it down to a very specific region is needed for DNA binding, and it depends on exact. So up here, they find this to be the DNA binding domain, and this to be the activation domain. And you can get very specific. You can narrow it down to specific amino acids. So what that tells us is that transcription factors are modular. All that means is if you've got a protein, and this represents a protein sequence with an N-terminus and a C-terminus, you can identify specific regions that do one thing and other regions that do other things. And so maybe this is the DNA binding domain, this is the activation domain. There could be other domains. There are certainly other potential domains, but at this point, let's just think of these two. This is just showing different transcription factors, saying you can have, these domains can be organized differently, you can have multiple of each, that kind of a thing. These are just examples of different transcription factors. But the idea is that this is evidence that transcription factors are modular, meaning there's different modules that do specific things. This is our DNA binding module or domain. This is our activation module or activation domain. And in fact, you can take this out and put it anywhere in any protein, and it'll make that protein bind DNA. So transcription factors are modular, meaning you have a protein. So let's say this is a gene for the protein. You make a protein. It folds up into its proper structure. That's not prime. The N terminus and the C terminus. And somewhere in there, let's say this is our DNA binding domain. How do we know? Because we delete that region of the protein, it doesn't work anymore. And let's say this is our activation domain. And the idea is that on a target gene, this protein, so there's going to be some sequence in this protein, and our protein has a DNA binding domain and an activation domain. This activation domain interacts with all these other proteins that cause transcription to occur. Okay. This is just a picture. So after they've done all these experiments, this is a super close-up of a what they call an enhanceosome. And so this is a little bit confusing. So this isn't all that important, but since the slide's in here, I'll just talk about it. So what they're saying here is they're taking a close-up view of what's happening in an enhancer. So you have an enhancer way down here, and it's going to interact with all this stuff. And before we've drawn it with a big loop, they're saying there's a little loop right here with a bunch of proteins bound to it. So it's not just one protein binding the enhancer, and that's the enhanceosome. I think that's what they're saying. Yeah, I think so. And then before we drew that everything looped, so now we're going to say here's the enhanceosome is like here, and all these proteins are bound to it. And then you got your mediator, and then everything else down here. That's all that picture is showing. You don't even worry about that one. All right, so here's an overview. This is showing that we start out with condensed chromatin, and then we can loosen it up, and then there's going to be certain cis-acting sequences that are in here that get bound by proteins. And the way they've drawn this is they've drawn their DNA binding domain and the activation domain. The activation domain interacts with the mediator, et cetera, et cetera. We talked about that already. We'll skip this part. This isn't all that important. This just shows you that these are all the different general transcription factors and how they bind in the order they bind. But that's too much detail for now. Just know that TF2D contains the TATA binding protein. It binds the TATA box, and it kind of starts the whole process. That's basically all you need to know. All right, so I think we'll stop. Well, let's take a look at how far we've gone here. Doesn't tell us. All right, we're going to keep going just for now, just because I just want to talk about this one last, because this is very related to the other thing. So there are some transcription factors that are turned on directly by hormones, by steroid hormones in particular. And these are pictures of steroid hormones or steroid-like hormones. So cortisol is a steroid hormone, and this is the basic structure of a steroid. And estrogen, testosterone, cortisol, things like that are steroids. These things are not steroids, but they're other hydrophobic or nonpolar hormones. And so they can go right straight through the membrane. So the way these things work is if you have a cell, these hormones can just go right straight through the membrane, and then the receptor for those proteins or for those hormones is in the cell. So that's a protein. So how do these things work? So here's a picture of some of these transcription factors. So these are hormone receptors that act as transcription factors. So if we go back up. So I just want to show you quickly how these things work. So here's a nucleus with a gene. It's got some cis-acting sequence. There's a protein in the cytoplasm, and there's some kind of a hormone out here that will go through the membrane and bind onto this receptor. Then this goes into the nucleus and binds onto the DNA. And there's another protein that binds onto this, which is an inhibitor that keeps it out of the nucleus. But when this hormone comes through the membrane, binds onto the receptor, that allows the receptor to sort of let go of this inhibitor, so this stays out here. Then this goes into the nucleus, binds onto the enhancer or promoter element, and activates transcription. So that's how these things work. Let's go to the next page. And this is a bunch of examples. This is showing the sort of protein sequence of each of these things. And each of these things has, this is just to show that there's other types of domains. So we got our DNA binding domain. There should be an activation domain in here. They don't really show it. Let's say that this is the activation domain. And then they've got a domain that is the ligand binding domain. So when I showed that if you've got a cell, this receptor is inside the cell, this hormone goes into the cell and binds on. Another word for this hormone is ligand. Some people say ligand. And then, so there's a domain that's part of this protein, which is the ligand binding domain. So and then this is these are the elements. So when we draw a sequence and we say there's a sequence element in this gene that responds to a particular hormone, then that's the response element. So we have glucocorticoid response element, estrogen response element, vitamin D response element, thyroxine response element, and this is retinoic acid. So these are all examples of these hydrophobic hormones that turn genes on by activating transcription factors, and they bind to specific sequences, and those things are called response elements. Here's an experiment that shows how people identified this domain. So they took beta-galactosidase again. They didn't quite use it as a reporter gene this time, but all you need to know is that wherever this light stuff is, that's where the beta-galactosidase protein is. They actually use an antibody against the protein. This is one of those hormones called dexamethasone, and what they're showing, the bottom line is that when you add, so if we go back to this picture, when you add hormone, the receptor goes into the nucleus. So they're showing, that's what they're showing in this experiment, is that when they add hormone, in this case, for example, over here, this is the nucleus. So the protein has gone from the cytoplasm and gone into the nucleus. Beta-galactosidase by itself doesn't do that. It stays out in the cytoplasm. But if you connect it up to various parts of the glucocorticoid receptor, it goes into the nucleus. And it turns out that if you just add this one little bit, it goes into the nucleus. So you can do that experiment. And that's how these different domains were identified, by taking these domains and hooking them up to some other protein and showing that those domains could cause the thing to go into the nucleus, but they only do it in response to hormone. So in the absence of hormone, they stay in the cytoplasm. You add hormone and it goes into the nucleus. So that's that experiment. And these are just close-ups of that experiment. This is kind of what I just drew. So you've got this receptor. You've got a ligand binding domain, a DNA binding domain, an activation domain. The hormone binds to the ligand binding domain. Here's that inhibitor I was talking about. Causes this to go into the nucleus. The DNA binding domain binds a response element. The activation domain is going to interact with the megablob, TF2D, RNA polymerase, transcribe the gene. So that's just sort of an example of how you can turn genes on and off. There's a picture of the mediator complex. Just memorize all those proteins, no big deal. Doesn't really matter. Just means there's lots of proteins. It's called the megablob. So that's what that thing looks like. And I'm trying to advance this thing. There we go. And that's just everything sort of put together again. So this is showing, once again, up here we're going to have maybe over here, maybe here, an enhancer, DNA binding domain, activation domain, megablob, mediator. Don't worry about those. These are general transcription factors. All these TF2 things are general transcription factors, and they help to recruit RNA polymerase and then transcribe the gene. All right, so we'll stop it there, and then we'll look at some chromatin experiments, and then that'll be